Good morning, everyone. Thank you for coming. I hope this doesn't seem too formal, but we decided that I would introduce my lovely students since this is going to be our final day together. So we have Owen Bearman coming to us from Saguaro High School. He's going to be a senior. We have Nathan Margolis coming to us from Pasadena High School, or PHS, who's going to be a junior. And then finally, Hailey coming to us from Blair, who's also going to be a senior. So as Mitch had mentioned, we're all coming to you from the Bronner Lab. I'm Brittany, I'm their postdoc mentor. And it's been a real pleasure to have them in the lab this summer. And over 6 weeks, I've been so impressed with what they've learned, what they've been able to accomplish, and so many things that I've been able to take away from them. So I'm really excited for everyone to see what they've been up to this summer.
Hello everyone, my name is Nathan Margolis, and I'm going to be discussing a little bit about the peripheral sensory structures of the zebrafish. So, across vertebrates, the process of embryonic development is highly conserved. The peripheral sensory structures in zebrafish arise in two embryonic stem cell populations, the cranial placos and the neural crest cells. For the past 6 weeks, we were focused on two major sensory structures, the inner ear and the lateral line system.
We used zebrafish as a model system for our experiments, and this is zebrafish are the ideal model system for these kinds of experiments for a multitude of reasons. The first being that they develop rapidly and in large groups, which makes them pretty ideal for sampling and experiments. And also, you know, if things go wrong in the experiment or they die, they can be easily replaced by other zebrafish. As well as zebrafish are vertebrates, and the process of embryogenesis is the same or similar across all vertebrates. So it can be modeled in comparison to other vertebrate species. And lastly, zebrafish are easy to manipulate genetically. We specifically use the transgenic line 61B GFP to pronounce the certain notes in our imaging.
So as Nathan said, the two structures that we were most interested in were the inner ear and the lateral line systems. And in zebrafish, the cell that's most prominent in dictating the sensory activities of zebrafish are the hair cells. So if you look on the far, your right image of the zebrafish, each of the L1, L2 are the neuromasts. And from each of these neuromasts stem these hair cells. And so when these hair cells are distorted, it can elicit a neural response and help the zebrafish navigate, and specifically in the inner ear, help them hear. And so our objective, or I should mention that the leading cause of hearing loss in mammals is the loss of these inner hair cells. And so what's interesting about zebrafish is that these hair cells regenerate, but in mammals they don't. So by selectively ablating these hair cells, we can design an assay that can analyze the regeneration processes of these hair cells. So that is our objective, to design an assay to assess hair cell regeneration in zebrafish.
So for experiment 1, we have different samples of time courses of these zebrafish embryos. We have 24 hours past fertilization and 48 hours past fertilization, 72 hours and 5 days. And we did this so that we could choose what time course was the most ideal for the ablation experiment. And we chose this because we wanted a sample that was reading positive and bright in the GFP, glowing, and also had fully developed neuromasts. And we ended up picking the 72 hours past fertilization sample because, as you can see in the 24 and 48, the neuromasts, which are these guys and those guys, are much less developed and much less shapely, whereas in the 72 hours, they have fully developed their shape and are glowing much brighter. And we didn't use anything more mature than 72 hours past fertilization for a number of reasons. One being that the longer that they mature, we found that the more that the muscle tissues started emitting the 6-1b GFP signal. And you can see it in this 5 days and even in later samples that like the muscle tissue is very pronounced. And this kind of convoluted the images and made it harder to observe the ablation, as well as just older, more mature embryos were more difficult to work with. And the older they are, they experience bleaching on occasion, which also convoluted the image.
So what you're seeing here is the copper treatment we did of the 72-hour past fertilization embryos. It's been shown that in small concentrations, copper sulfate can selectively ablate hair cells without damaging or compromising any other non-selective structures in aquatic vertebrates. For this experiment, we reviewed the ablation of the hair cells in zebrafish at 4 different concentrations, control being 0, 1 micromolar, 10 micromolar, and 50 micromolar of copper sulfate. And we used 2 different time stages post-treatment to observe the recovery of hair cells after being exposed. We selected the 50 micromolar copper sulfate solution as the hairs were effectively ablated while maintaining other non-selective structures. So as you can see, it gets brighter the further you go down the control to 1 to 50 micromolar. And that just means the hair cells are more visible and we can get a better image of them when we do IHC staining, which is what Hailey's going to talk about.
Okay, so like Nathan said, we stuck with the the 50 micromolar concentration of the copper sulfate to ablate these hair cells. And by using IHC, we were able to selectively label some of the cells in the zebrafish embryo. So here, the green color kind of represents the GFP labeling where the neuromasts are. The red color represents the tubulin labeling, and it's like where the hair cells are. And then the magenta color is our pH3 biomarker, kind of showing where the what cells are in the late G2 to mitosis stages, so those that are regenerating or proliferating. And so the purpose of this part of the experiment is to further assess hair cell regeneration processes. And so we have these time points throughout the copper treatment. So the 0 right here is before any treatment. So you can see that the green and red colors are kind of a little more organized. So the hair cells are in coordination with the neuromasts. And there is some regeneration, but we won't see that till later. And then the T0 is 2 hours after treatment. So we can see that the red and the green are a lot more disorganized, which is good. It means that the ablation experiment worked. And so we can see that it's a lot more disorganized as compared to the 0 time point here. And as we move on to the next time point, which is assessing like its recovery processes after the 2 hour or it's 6 hours post treatment, we can see a lot more regeneration happening with this pink biomarker here. And the red and the green are a little more organized so that the hair cells are being are regenerating well. Yeah, so and it kind of reaches its peak regeneration here. Oh.
Let me just put that back on first. So we have successfully developed an assay to ablate hair cells in zebrafish and assess their regeneration. We use copper sulfate to ablate the hair cells and IHC immunohistochemistry staining to visualize their regeneration.
And we can utilize the success of this ablation experiment to possibly apply it in broader landscapes and discover other genetic mechanisms and physiologies involved in the regeneration of hair cells. that can aid us elsewhere in science.
So like I said earlier, it's interesting that the hair or inner hair cells of the zebrafish are able to regenerate as opposed to mammals. And so learning about their regeneration processes can help us to potentially find treatments for hearing loss in humans in the future. So we included some famous figures who were deaf. So this is a time for Q&A. If you have any questions, please feel free to answer. I mean, ask. And we will answer.
I have a question about your slide with all the different colors on it. I don't understand the coordinates of that. Is that one zebrafish you're looking at or?
Yes, so this is one zebrafish. They kind of are images of different areas of the zebrafish. So here, this is like the eye, right? This is the head of the zebrafish. So each of these little green, like floral kind of looking things, these are the neuromasts, right? And then the red sort of lines here are the hair cells stemming from these neuromasts. So when the hairs are like moved throughout the or like what's it called, distorted, they will elicit like a neural response causing, or allowing the zebrafish to navigate waters and such.
And this was viewed looking with a microscope?
Yes, this is us under a microscope. I believe these are all under the 20x objective. And yeah, so if you look at this one, oh, just kidding. Yeah, so these are all under the microscope. Yeah.
Thank you.
Hailey, can I ask your question about future applications?
Yeah, of course. So like one of the questions that we had was like, how far in the future do we see these, this research that's being done being applied to potential hearing loss treatments. So there was a study done in 2010 with adult monkeys, and they used the AAV virus, the adeno-associated virus, to perform gene therapy on these colorblind monkeys. And so they were able to restore color vision to them. And so, with that knowledge, maybe we hopefully can see this type of research being used in the future for hearing loss treatments. Any other questions?
I think that's it.
Thank you.